Thank you very much. A pleasure to be here this morning. So as Roger said, I got my PhD here a few years ago. Maybe I shouldn't say how many. So my PhD is in computer science, and about 15 years ago, I became fascinated with the notion that we might be able to program cells as easily as we program computers. And in some sense, that's what synthetic biology is right now. So I'm going to talk about that and give you a flavor for what's going on. So what I'd like to do is emphasize that you should ask me questions. Ask me questions throughout. Presumably, people here have different backgrounds, different knowledge, and so on. So ask me any kind of question. I will try to cover things from the ground level, but sometimes there's vocabulary that you're not familiar with, or something like that. So please ask me whatever kind of question that you'd like. When I got started in synthetic biology, we're looking for some kind of an analogy. Anybody seen this movie? Yeah, cute little love story, right? But the point is, we're looking for an analogy to try to help us kind of better understand, get better formulate what synthetic biology is about. And robotics was a nice analogy. Since then, there's been many other kinds of analogies: bridges, cars, planes, boats, radios, even bridges. So the reason I like this particular analogy of robotics is this notion that we have a robot that's a physical entity that we engineer to interact with the world. Okay, and I think to a large extent, that's what we're trying to do with biological cells. So when we think about a robot, we often think about this physical entity as having something that has sensors that can sense what's going on in the robot, what's going on in the environment, then takes that sensory information and processes, makes decisions, and those decisions then control the actuation. So it can modify what the robot is doing, or it can modify what the environment is doing, or affect itself. So we can basically make these corners be more rounded. We can label this as biological cell. To a large extent, that actually exemplifies what we're trying to do in synthetic biology. So rather than doing it with electronics, so I guess we're ignoring that. Okay, we gave up on that. Yeah. So my PhD in computer science, I guess, didn't really help here. So that's why I switched to biology. Using DNA to make circuits as opposed to using electronics. Okay, so we actually build pieces of DNA, and I'll give you a flavor of what that means. We put that in the cells, and a flavor of what the DNA actually contains, what the instructions are, and then as a way to control what it's actually doing. So in terms of the cell, so we look at the cell. What are the things that we control in the cell? So we want to try to regulate activity in the cell. So gene regulation, protein activity, translation, protein-protein interactions, get the cells to synthesize various molecules for us, sense, again, chemical, light, forces, be able to control the physical aspects of the cell, motility, growth, transport of various things, and also always think about the cells not being an individual entity, but something that really interacts with other cells nearby. So it's part of an ensemble of a population of cells. So we want to get these cells to somehow communicate with one another. And so these basic properties then allow us to start thinking about applications. So what is it that we want to do with synthetic biology? So these are the various applications that are typically discussed. This is maybe a sample of the applications. So again, this notion of synthesizing molecules. So one of the more famous ones is artemisinin, which is a malarial precursor drug. Okay, so there's actually now a company that's focused on making this very cheaply. Bioenergy production received a lot of attention. So can we get cells to produce energy for us, clean energy for us? Toxin sensing, environmental remediation. So in the Gulf spill, for example, it would have been nice if we were able to engineer bacteria that would just go into the Gulf and clean up the oil spill, or maybe even aggregate the oil for us and give it back to us and say, here you go, don't worry about your spill, kind of thing. It turns out that, interestingly, about 20 to 25% of the oil spill was actually cleaned up by bacteria. Okay, but these were naturally occurring bacteria. These are not ones that we engineer, and these bacteria were actually able to really help out. What I'm going to do today, towards the latter portion of the discussion is get into a little bit more detail about biomedical applications. Okay, so I'll talk about cancer therapeutic, I'll talk about building tissues by design, and tissue homeostasis for beta cells. Okay, so in terms of my own lab, we focus a little bit more on these types of biomedical applications. Okay, so in terms of kind of defining synthetic biology, I guess everyone that you talk to in synthetic biology is going to give you a slightly different flavor for what it's about. But to some extent, the goal is to design sophisticated biological systems. To really, if you think about genetic engineering that started in the 70s or so, the emphasis there was how do we take a gene, perhaps a couple of genes, overexpress them in the cell and try to modify what the cell is doing. In synthetic biology, we're thinking about systems. We're trying to understand how to go from the individual gene for a couple of genes to really create large-scale genetic systems in kind of in a programmable fashion. And it's similar to the transition in a sense that systems biology provided. This notion that we're not looking just at an individual gene, individual protein, or even a portion of a pathway, but really about thinking about the systems as a whole. In systems biology, the emphasis is on analysis, and here the emphasis is on creation. Okay, so being able to think about, I want to have this particular behavior, how do I create something like that inside a living cell? And so what we try to do, many of the folks in synthetic biology are actually people that come from other disciplines and not necessarily biology. Actually, it's kind of surprising that most people are really not from the biological field. Okay, many people come from different engineering disciplines. And so what people bring to the table are these notions of how are we able to build computers, and I mentioned computers, cars, robots, and so on. And so these engineering practices have really helped us be able to handle the creation of these sophisticated systems. Okay, so this notion of having libraries of parts, testing them, having modules, using computer-aided design. So we're trying to bring a lot of these principles from the different engineering disciplines. Okay, but at the same time, and this is actually overlooked to some extent, is we have to understand what's different about the biological substrate. Okay, not really take it for granted, not oversimplify, not saying, look at a cell, oh, that's exactly like a computer. And then if I know how to program, in Java, that means I know how to program a cell. Sometimes people actually, in a sense, use, think like that. And it's really important to understand what is different about biology. So for example, if I say, I want to take this component, this genetic component, characterize it, this other genetic component, characterize it, and put them together. Can I predict exactly what would happen? Okay, so it's 1 + 1 = 2. And even that simple notion is something that still really hasn't been resolved. Even taking the very basic parts and putting them together and being able to exactly predict what happens. Okay, which you imagine really would be necessary. Noise is a big issue, right? So biological systems have a lot of noise that we have to deal with. Cellular context. So when we build a genetic circuit, the operation of that genetic circuit really depends on where it is. Where is it operating? I mean, even if you put it in one type of bacteria and slightly change to a different type of bacteria, you can get vastly different behaviors. Okay? It's certainly quantitatively. And so it's important to understand that. Many other things, so I'm not going to go mutations we have to worry about, cell death. You have to worry about, so you really have unreliable components you have to deal with. And usually when I say this, people say, oh, well, you're using Windows. So, the situation is not that much different. It dies on you all the time. By the way, I actually just bought a couple of Macs. So I don't know, we'll see if my situation gets better. So I'll report on that in a few months, I guess. And so the bottom line is that we need to understand what works in different engineering disciplines and what is different about the biological substrate, and create a new engineering discipline. Okay, and that's what synthetic biology represents. And it's also important to understand that it doesn't have to be exactly like nature. Okay, so nature, really, there's a process of evolution that resulted in the genetic makeup of all living organisms, right? At least for now. That might change in 10, 20 years. That's actually not true. Maybe in the labs, all those organisms are not really evolved by nature anymore, right? But so the process of the evolution, what happens is typically is there's some mutations going on, some genetic recombination, and you get a new organism that somehow is a better fit. It has better fitness in a particular context. Okay, so those are the rules. That's a rule by which evolution operates, and by the rule by which you get new versions of the DNA. And in synthetic biology, we really don't have to do that. So for example, we can make intermediate versions of these organisms. They're actually not robust, not very functional, don't really work very well, and learn from that. And then kind of leapfrog and make some drastic changes. Okay? So the other thing that we might want to do, that nature typically doesn't do very well, is make things that we understand. Right? So we'd like to create systems, maybe they're not completely optimal, but we'll make version, a graduate student will make version 1.0 in order for the next graduate student comes in to be able to easily understand the system and make version 2.0. Right? So the notion of understanding is actually an important notion in synthetic biology, not so much in nature. Okay? So we really have to think about what are the rules that would work quite well for us as an engineering discipline, and not just one ad hoc construction of a single genetic circuit. So what I'll do today is I'll quickly go over DNA synthesis and assembly just to give you a flavor of what the DNA is all about. And really this has been a really important component, the driver for synthetic biology, for the progress in synthetic biology. So the similar kind of Moore's law driving electronics, advances in DNA synthesis and assembly have really been critical. Then I'll go into some basic design. So what is it? Parts, then taking parts, understanding what the parts are and building them and making modules out of them. Computer-aided design. So this is important. This is an emerging aspect of synthetic biology. And then I'll briefly go over, what can we do? Right, so besides building parts and modules, what is it? How do we think we can actually really radically change different fields? What is this notion of being able to program something? How does that change what we can do with respect, for example, for cancer in tissues and in diabetes and so on? Okay, so in terms of DNA, so again, I want to emphasize, please, don't be shy, ask me questions. So now we're going to, in a few slides, we'll get a little bit more technical. So I do encourage you to ask me questions, and it doesn't have to be, it could be philosophical questions too. I can, I take those, or I can point to somebody else to answer them. We can have a discussion. So DNA synthesis, what are the scales here? So we often think about the size of the circuits that we're building, and so there's the notion of building individual genes. That's a typical scale that we think about. Start out with genetic engineering. I'll give you a couple of examples here. In terms of synthetic biology, we usually think about building genetic circuits. So that's the typical range that we think about, kind of a modular circuit to build. And then you can start thinking, well, beyond that, if we had 10 to the 5, less than 10 to the 6 bases, what can we do with that? And that's a region where you can already start thinking about minimal life. What does it take to create a piece of DNA that can actually support a living organism that would be at least somewhat independent? Yes?
How do you build a genetic circuit, and what are the principles in building DNA?
Yeah, so great question. So ATGs, the ATGCs are the components in the genetic circuit. They're basically the alphabet, like we have zeros and ones in computers and so on. They're the alphabet that we use, and then we arrange the ATGCs in a particular sequence where these sequences, portions of these sequences, encode particular function. Okay, so and I'll get into that in more details. But if you look here, for example, this represents a circular piece of DNA. That's a genetic sequence right here. And this portion right here, for example, represents something that's called promoter. And I'll go back and describe that. That promoter is a sequence of DNA that regulates the production of a protein. Okay, so this promoter here regulates the production of this protein right here that's encoded in this yellow-orange. Okay, and so a genetic circuit typically consists of these promoter gene pairs. Okay, so we're going to have promoter gene here, promoter gene here, promoter gene, and something that actually replicates the DNA. Okay, and so that's how we encode a function on DNA. Is we typically have these promoter gene pairs, and they result, when you put this into a cell, this results in the production of things like messenger RNA and things like proteins, which then are the things that actually really perform the work inside the cell. Yes?
How do we ascertain that the functions are sequential or not?
So you can say they're not. So we'll resolve that question, right? The function is not sequential. Okay? When you build a piece of DNA, it's sequential, right? So there's a particular sequence here, and the order, the actual letters that you have on there are very important because that's the thing that actually encodes the function. But when a cell looks at this, it doesn't care too much about the order in which this comes. There's all kinds of, so I don't want to get into all the the complexity, but structure matters. So how the DNA molecule is folded may matter. Cells sometimes make modifications to the DNA to make it less accessible, more accessible, and so on. But to first approximation, it really doesn't matter what the order of the regulatory elements is. And everything, if you think about it, everything kind of happens in parallel. Okay, so it's going to regulate this in parallel to regulating this. Okay, now when the order matters, whenever it makes this particular protein over here, so there's a molecule that binds to the sequence right here, and then sequentially goes over these DNA bases, and then makes a protein. Okay, so it's kind of confined to, yes?
How do you debug the bug?
How do you debug the bug? So mostly what you do, the easiest way to do that is make fluorescent proteins. Okay, so that's normally what's used at the basic level, is you make fluorescent proteins and they indicate what's going on. So that's kind of your, debugging statement, is to make fluorescent proteins. However, that's only useful for certain things. So sometimes if you have a circuit right here that makes that has some metabolic activity, then you want to monitor what small molecules are being made by the cells. So you have to use different things like mass spec. You have to use different pieces of equipment, right? Sometimes you would look at the morphology of the cell. So if you have a stem cell and you program it to differentiate to a neuron, for example, you can look under the microscope and see what's changing. You can use antibodies that are fluorescently tagged to find out whether that cell now has features on its surface that would be indicative of a neuron. Right? So there are various ways by which you do the debugging. Okay? All right. So great. So please do continue to ask questions. It's a lot more fun for me too, because I kind of know what I'm going to say. So these interactions are great. So this is the minimal life, and now we're also starting to think about rewriting genomes. Okay, so this is on the order of a bacterial genome over here. We starting to think about things that are slightly larger, like yeast, and eventually, can we write whole human genomes? I'm not going to guarantee that I'll be around for that, but certainly the technology is moving quickly. I will get back to this. So genetic circuits. We know the basic principles. Genomes, our ability to rewrite genomes is very, very early stage still. Okay, and I will get back to that point. Okay, so in terms of the technology that's driving it, I mentioned this notion of synthesis. So if you look at sequencing, this is always a good comparison. So sequencing costs are just dropping dramatically. Okay. So this is years versus the cost to sequence a DNA base. And then this is the cost for constructing short oligonucleotides. So those are the basic elements. Short oligonucleotide, this is single-stranded DNA that's typically less than 100 bases. Okay, so that's the foundation. And that's the foundation for building double-stranded DNA that's longer, as you start with short oligonucleotides. Okay, so this is the cost that's going down, and this is the cost of gene synthesis. This would be double-stranded DNA. Something that you might notice here that could be a little alarming is that the cost of oligonucleotide synthesis is dropping, but not that fast, and this is dropping faster, but there might be some intersection point which would be a problem. Right? So double-stranded DNA in principle shouldn't cost less than producing single oligonucleotides, right? You would hope. So we're potentially running into a problem here. So why is that actually not a problem? So I get to ask you a question every once in a while. Not that often. And by the way, the answer is on the slide. But how can that be that we look at this and we don't get, I mean, we get a little concerned, but we don't panic and, short stocks of synthetic biology companies.
It's 10 cents per.
So this is about 10 cents per base to synthesize. This is actually, I think this is about the right number back then. So these ATGCs, you can buy them. You can go online and you can buy an oligonucleotide, and you basically just type in the sequence. And for at that time, that's 10 cents a base. I think it's a little lower now, but that's you would get even next day if you pay maybe a little bit more. But 2 days later, you get a tube that contains lots of that molecule that you specified, typically under 100 bases or so. Actually, when you build them, that's typically under 60 bases. When you get a little bit longer, they get more expensive, right? And so then if you want to build a new piece of DNA, then you want to take a lot of these short oligonucleotides and kind of stitch them together to make double-stranded DNA that's longer. Yes?
How are they able to create the short nucleotides?
Okay, so good. Without going into too much detail, what they do is they tether something to the surface, and then they wash one of the letters over it, and it basically is ligated to the thing that's tethered to the surface. So if you want to have ATTGG, then what you do is you first wash A, and then you fix it in there, you cap it, and then you wash the A away, and then you put in the next letter and the next letter. So you're building it one letter at a time. You're basically expanding the oligonucleotide one letter at a time. And then when you're done, you cleave it from the surface. And then you put it into a tube, and then you put it in FedEx or something. Right? So you build it one letter at a time. So I specified something, lots. Okay, so I'll give you the answer, I guess. So here I didn't specify how much I'm giving you. I told you that I'm going to give you a sequence of DNA, but I didn't tell you how much. So I didn't tell you how many molecules do you get. So normally you would get a tube. So a tube contains lots of molecules, right? And I said, that's the cost of getting that particular sequence, 100 bases or so, right? You get many, many copies of those hundreds of bases. So the people have noticed that, and they said, well, rather than giving us lots of these molecules, what if we had this mechanism where we can print many different versions of 100 bases, where each version you just get much less of it. Okay, so if you kind of flip that around and you say how many bases can you synthesize for $1, now if you look at oligonucleotides, that's going up exponentially, right? So it's a very different way of thinking about it, right? So if you can minimize how much you make of any particular sequence, and you have for example, lots of these sequences around, you can have 1,000 of these sequences on a single chip that you make. You can, maybe 1 million of these sequences. Then the cost, how many bases you can make per $1 increases dramatically, and then that actually gives us hope. So if we don't need to use a lot of these oligonucleotides to create these double-stranded DNA, right, to create even a single copy of that double-stranded DNA, then we're in good shape. Okay, let me point something out. Once you create a single molecule of DNA that's functional, that has the right properties, you can take that single molecule, put that inside a bacterial cell, and then give that bacteria a little bit of sugar, and grow it at the right temperature. And overnight, you're going to get, tons. You're going to get billions and billions of bacterial cells overnight, and each one of them is going to replicate that DNA for you. So you have a very cheap mechanism to start from a single molecule of DNA that has the right properties to many, many copies of it. Right? And so that's where we're getting this potential benefit of if we can make just a few of these single molecules, we can still create DNA, double-stranded DNA, very cheaply. Okay, so this is basically why our Moore's law for DNA synthesis is actually on a reasonable track. Okay, so that's with respect to the basic DNA synthesis capabilities. In terms of now, let's think about these plasmids that I showed you. So this is what's typically been around. This actually, again, this was started in the 70s or so. How many people here have done cloning, have created plasmids? Anybody here? Okay. You did it this way? Okay. So this is old school way. Hopefully when you go back you won't have to do it again like that, right? So this has been around. This has been around since the 70s, and the idea is you have if you want to make a new piece of DNA, what you do is you find these restriction enzymes. These are proteins, and they're going to cut. These are names of it. They're going to cut in particular places, open it up, and then you basically do a lot of cut and paste operations, and it's lots of fun, right? And you wish you were doing it all the time, right? Yes, that's a major agreement. And so this is the way DNA synthesis has been done. But another mechanism to do it, so as you can imagine, this is pretty laborious, right? And so another mechanism is this notion of if we want to create something completely from scratch, how do we do that? Right, so we want to have a completely new sequence, a completely new gene. Okay, so what you do is you create these oligos that I just discussed, and so they can be 40 mers, they can be longer, they can be 100 mers long, and so you pull them together, and then they're going to have these mechanisms where they anneal to one another. Okay, so they're going to have some overlap between with Crick-Watson pair. So they're going to anneal to one another. They're going to create something like this. Okay, so now this is a chain. And what you can do is you can do something called polymerase chain reaction. I won't go into the details of that, but the idea is that you can then amplify the sequence. So even if you have very few numbers of these, you can use PCR to make lots and lots of copies of those. Okay, so you can use PCR on this, make lots of copies of those, and then put that into an existing plasmid. Okay, so this is from 1995, but this was actually even demonstrated in the late 70s. This notion of taking oligos, deciding here's a gene that I want to make, putting those in a tube, doing the right cycles and so on, and then creating a gene or a piece of DNA that may never have existed in the past. Okay, and so that's the way to create to create small genes here. 1995, 0.9 kilobases. Okay, I want to point something else out. So in synthetic biology, this is actually my former advisor at MIT. A little bit after I left, after I got done with my thesis, he came up with this notion of biobricks. Okay, so this has been one of the basic building blocks in synthetic biology, and this is this notion that we want to start. So I showed you how to create an individual gene, but synthetic biology really focuses on the creation of systems of genes. Okay, so he came up with this notion of encoding, for example, a gene, and now it's called a biobrick, but then having a mechanism. So let's look this is a biobrick, okay, and it has these restriction enzymes over here. And the critical thing about this biobrick is that you can always take this biobrick, and without thinking too much, you can put two biobricks together and make a composite biobrick. So if you wanted to create a long sequence, this is what you do. So you take, for example, these 8 biobricks, and you put them together in a hierarchical fashion. So you do cut and paste, cut and paste, cut and paste, hierarchical fashion. And the property here, that every time you do a cut and paste operation, it's always a biobrick. So it always contains, so whenever I combine A and B, then I have an A-B pair over here, but it still contains the right BioBrick cut sites on the outside. So that's a particular property of how this is made. So you can always take two pieces, you cut them in the right way, you ligate them together, and now you have something that contains both pieces. And the cool thing about it is that now that piece can be used in further assembly. Okay, so that's the BioBrick Assembly mechanism. So that's been important as a foundation for the field. And it looks pretty nice, so that allows you kind of to say, I want something that A, B, C, D, E, F, G, H. So let me find these basic components, and then here's a plan for making this. And this is actually very easy to come up with this plan. Okay, there's not the design phase of actually making the DNA is rather easy. Okay, the other thing about it is that it's not just a process. It's also the information of thinking about things. It's the way you view these materials. So now you start thinking about these parts, and you start thinking, well, if I have A, B, C, D, that might be a module. You know, it's a BioBrick, it's a composite BioBrick, but it's a module that might contain interesting information in it. So I'm going to think about that module, I'm going to describe it, I'm going to characterize its properties, and I'm going to put that on the website. There's actually a registry of standard biological parts that has about 7,000 of these. So if you want to build something, you go online to this website and you say, I want to be able to build this fairly sophisticated system. Let me go shopping and go online. Kind of like when you go to Radio Shack. People, I guess, don't really do that anymore. Back in the day, they used to do that. So you say, I want to build some composite system. Let me just go online, and I want to take this part, this part, this part, this part, and it's say, ask the registry, can you please give me these parts? And then you get them in the mail, and then you put them together, and you create your new system. Okay, so it's both a standard way of assembling DNA, as well as, perhaps more importantly, an information source that contains all of these pieces, which then allow you to very quickly think about building more and more complex systems. Yes?
So does each oligo, does that represent like a small portion of a protein or a protein?
Yeah, so each oligo would be, for example, a part of the construction of a gene. So in order to make a new gene, so for example, this might be a way for you to make a completely new gene. So you say, oh, I'm going to have this gene that has this really cool function because it can metabolize something in a very efficient way. So you say, okay, I'm going to make these oligos, so get them synthesized, and then I'm going to put them together and make a new gene right here. And so the new gene could be, so typical sizes are about 700 bases, 1,000 bases, could be more, 5,000, could be, but those are the typical sizes. And then you make that new gene, and this would be, so that could be your gene. Okay? Yeah.
So why is two restriction sites before and...
Yeah, so the two restriction sites allow you to cut things so that they come before or after. Okay, so what you do is if you want to put together two BioBricks, then you will say, for example, I want this one to come before this one. So what you do is you actually cut, so let's say you want A before B, you cut the one in blue here, and you cut the one in red. Okay, so you cut the blue, you cut the red, and the cool thing about it is then the blue and the red come together, they can come together. They're special in the way that they get cut. Okay, because of the actual DNA sequence, what happens is that you have these four DNA bases that get exposed from the blue, and they're compatible. The four bases over here with the blue are compatible with the ones on the red, and they ligate and they come together. Okay, and then what would happen? So you actually get the sequence yellow, red, A, blue. Plus, so that's the piece that you take, yellow, red, A, blue. You actually physically take that, and you put that together with red, B, blue, green. So again, you physically have these two components that you cut with these restriction enzymes. And then the thing that's really important is when you put a blue and the red together, that's no longer a site. So it's a blue-red mix that's no longer cuttable. And that's really the trick here. So when you put yellow, red, A, blue, red, B, blue, green, you get yellow, red, A. In between the A and the B is the blue-red cut site, but blue-red is nothing. It doesn't respond to anything. And then you have the B, blue-green. And so that's the thing that actually allows you to put these things together. You have this mixed cut site, no longer responds to restriction enzymes, and you can build, and the part that you created basically has the same restriction sites as the parts that you started with. Yes?
For the next step that you want to combine A, B, and C, D, then if we want to cut blue and red, then the site between A and B doesn't move?
Exactly. Yes. So why? Because the restriction site is 6 bases. Okay? And so what happens is, so let me say you have the blue and the red. So if you think about the blue and the red, so they recognize 6, this is double-stranded DNA. Okay, so this represents the 2 strands of the DNA. This is 6 bases, okay? And then the way it gets cut is like this, okay? So what you're left with is this sequence right here, okay? And then, let me make sure I match it up. And then this sequence right here, okay? So you get this blue, you get this red, they come together here. This is compatible with this, but the key here is let's call this B1 and B6. So these are the positions, and this would be red 1 and red 6. So when those two come together, it's B1, right? And then B2, 3, 4, 5, 6, and then R6, right? And so what happens is you get a mix of B1, R6, which no longer matches either B or R. Yeah. That makes sense? Yes?
What happens if the restriction site performing genes?
Great question. So one of the restrictions about these genes is that you cannot have those restrictions. So that's a great question. And so that is definitely an important aspect. And so oftentimes you actually have to synthesize the DNA. So you have to let's say this gene and this awesome gene, and you know it's going to cure the world of hunger, but it has the restriction site on it like this. And you're like, oh my God, I can't cure the world of hunger. So what you do is you have to modify that sequence. So it no longer has that same restriction site. And so it's not the end of the world, because with typical genes, so for most of the time it's not the end of the world. So for a typical gene, what you can do is you can substitute a slightly different sequence that would still encode the same protein. So people know about codons, right? So the idea is that you can use, typically you can find some mutation that would create still the same codon, the same amino acid, but would have to have none of that site. Now, it gets more complicated if you have a promoter. So imagine having a promoter where that sequence is actually essential. Then that could be a problem. And then you have to really think hard about that. Now, so that's an important constraint with BioBrick Assembly. Any other questions about BioBrick Assembly? Yes, and then I'll move on to the yes.
Which basically have combined different restriction enzyme, because for restriction enzyme the temperature, the buffer sort of requirements are different. So how we can?
Yeah, so good. So the idea is that we're always using just these 4. So these are in particular EcoR1, XBA1, SP1, and PST1. So those are the only ones that are ever used, and they have the exact same. So they were chosen very carefully so that XBA1 and SP1 are compatible, Okay, they're compatible in terms of ligation, but also all of these enzymes are compatible in terms of their buffer and restriction enzyme conditions. So same temperature, same inactivation. Yeah. So you always, so the nice thing about this, you always just use the same ones, but there are problems. So some of the things that we've mentioned here. So for example, one problem was that you restricted to a set of sequences. Now the other problem is that this, while in theory sounds pretty good, what happens if you have, 90% success rate? 90% success rate in biology is awesome, right? But when you start thinking about these, building these trees, 90% success rate is really a problem, right? Because almost any tree of any kind of interest is going to have a problem. And when I say problem, means that you just try to put these things together and they just don't want to come together. And then you're trying to figure out why, and this, it's just not working. And so this is kind of a limit in terms of sometimes our understanding of biochemistry and biology is that these things just don't want to come together.
So does it have to do with the reaction?
So there's something in that reaction, there's something in that, so as these pieces get longer and longer, it actually becomes more and more difficult for those pieces to come together. And so they're just, it could be that there's something annoying in this sequence right here that causes us to have some secondary structure that it just doesn't want to come together.
But even when you start from very basic structures,
Yep.
and if your reaction is not complete and you have some leftovers, and when you mix it further, they might
Okay.
something different and give you something which is undesired.
Yeah, so one of the things, so I didn't describe this in full detail because people are trying to move away from this. So I wanted to just, is that what you do if you put A and B together, what you do is you mix it, you put them together in a test tube, and then you typically put them into, you transform them into bacterial cells and you grow them such that each initial bacterial cell becomes a colony, and then you actually extract the DNA from that colony, and that should be just a single initial molecule that results in many, many copies. So a single molecule goes into a single cell and then grows into a colony which you can then extract the DNA from, and then you check it. So you do things, you cut it to see if it has the right lengths, you can sequence it. So before moving on from AB to ABCD, you have to verify that AB is in fact correct. And so that ends up being a huge factor in the delay here. So each step here is about, let's say if you're really good, 3 days. That's if you're not into sleeping too much. So you can do it in about 3 days. But that ends up, so again, that ends up, doesn't sound like much, but that ends up adding up. And then if you have an error rate along the way, that becomes really problematic, right? And so what you end up having to do, so if you have some error where you just can't put E, F, G, H together, regardless, so you can spend 2 weeks here, it just doesn't work, and then you have to give up. Then you have to basically reconstruct the tree in some other way. And so that's why it looks really promising, ended up, while being important, there's now transition. So I would say the people are still working with this. There are many. So for example, there's in synthetic biology, there's something called iGEM. People heard about iGEM? Okay, so the International Genetically Engineered Machines Competition, no relationship to GEM4. But there are now over 160 teams that compete in this from around the world every year. So these are undergrads, different schools, and basically each team of undergrads the key is just do something cool in synthetic biology. That's essentially the criteria. And so most teams still use this in iGEM. So most of the teams, but there's been over the last couple of years been a transition away from that. So that would be this. So maybe 5 to 10% of the teams have now started to realize that this is a problem. And while this is not still a part of the BioBrick Assembly, this is huge here. Okay, so what is this? So rather than putting 2 pieces at a time, so Dan Gibson, and I'll mention work that he did, he came up with a strategy of putting many pieces together at once. Okay, so now this is actually based on, there was some other mechanisms to put many pieces together. So his was some slight modification. In my lab, we use his mechanism. We use other mechanisms, something like Golden Gate Assembly, which is actually still has its benefits for putting many pieces together. But this has been huge. So the idea here is that you want to have all these pieces, and now what happens is you have this overlap region right here, this and this, for example. And you see there's slight differences in the color here. Now these are about 40 bases. So not 4. This is 4 bases. These are 40 base sequences. Okay, and the idea is that you basically expose. So they're double-stranded, and you have this enzyme that exposes these 40 bases. And so now, because they're 40 bases, they're much longer. They have a lot of specificity. Right? You can mix together 10 of them at the same time, and they find each other because of the specificity in the 40 bases, and they come together. In one pot, you can put 10 pieces together. You can actually put up to probably about 20 pieces together at once. And so that's made a world of difference in terms of what we do. So the idea is that you take all these pieces in one pot, you put them together, there's a plasmid backbone that you ligate them, and you get something like this. And this would be on the order, same time scale. 3-4 days to make something that's on this, at this level of complexity. So in my lab, we do this now routinely. We can create something that has about, we've done up to about 15 regulatory elements that we put together. So it actually consists of first putting promoters and genes together. And then, so each one of those counts as one regulatory element, and we can put about 15 of those together. This whole process is in 1 week. Okay, and that's really transformed our capabilities of what we can do. So this is the new way of constructing DNA. So when you go back, I don't know if you're still into cloning. Yeah. Do you do any Gibson?
I haven't.
Okay. You want to write it down? Okay. So at least one person will benefit from this discussion. But this has really been, a huge transformation in what's possible with DNA synthesis. And now, like DNA synthesis and assembly, so I'm showing you this, is now kind of the wild, wild west. I mean, it's just unbelievable. There's so much activity. People realize, oh my God, there's potentially other ways of building DNA. This represents now, hopefully by the time you get home, this would still be relevant. But I know of other mechanisms that people are putting together now. We have one to ourselves that might replace this too, within a year or two. So it's a really exciting area. It means that when you commit to a particular method of doing DNA assembly, there's a huge commitment in terms of the infrastructure. You have to pay attention and realize that next year there could be something else that just blows us away, right? And that that's the thing that's going on right now in DNA synthesis. And probably many of you have heard about this now. So this is Craig Ventnor, and Dan Gibson is the one that came up with Gibbons Assembly. He did it because he wanted to create, in the Ventnor Institute, create a synthetic genome. Okay, so what they did is they had this company called Blue Heron create these basic building blocks. Okay, so they created these 1 kilobase cassettes, and then they put them together in multiple steps, and they put them together 10 at a time. So they took 10 pieces, they put them together, and they took 10 more of these pieces, put them together, and 10 to the X grows pretty fast, pretty quickly, and they're able to assemble something that's about 1 million bases. Okay, I think this project was about $40 million, so it's still a little expensive. But this was, this is something that received a lot of publicity in 2010, the creation of a bacterial cell that actually is fully created from oligonucleotides that were created in the lab. Okay, so have most people here heard about this? Okay. And so this is, this is what's been created. So it's Mycoplasma mycoides, and it has a couple of changes. Now, something that I want to emphasize here, and then I'm going to switch to design, is that while we have the ability to create 1 million bases now in the lab from scratch, this doesn't represent a new design. This is basically some existing design that you make a copy of, and then so he put a couple of different genes in there. Okay, one of them for antibiotic resistance, and he also, Craig Venter, put his initials in different places on the chromosome too. Tells you something about, yeah, all right. But that was it. I mean, basically a copy. It's a functional copy, so it's a great achievement, but it is a copy. Okay, so in terms of DNA synthesis, this has been really a nice progression, culminating in this 1 million bases of a single DNA molecule. And so, kind of switching over to the synthetic biology aspect, we did a study a few years ago. Actually, it's still relevant. This is in terms of what's going on. Okay, this is in terms of what's been created in synthetic biology in terms of a new design. Okay, rather than making an existing copy of a pathway, put together new pieces in a way that's never existed before. What's been accomplished? Okay, so we did a survey of the literature of experimental circuits that have been made, been shown to function, and so 4 regulatory elements in 2000. So by regulatory elements, I typically mean a promoter and something downstream of that. So 4 in 2000 have been kind of a lull. This is a moving average. So then 3, 4, 5, 6 in 2005. Okay, so 6 elements put together, and up to even today, there's not been a publication that has more than 6 new regulatory elements put together in a single circuit, which is pretty amazing. Okay, so the bottleneck really hasn't been necessarily the synthesis. That's been, we're getting, let me rephrase that. That hasn't been the ultimate bottleneck. It's helping us a lot when we can synthesize further. But our ability to design has been also a huge factor in this. Okay, so I'll talk about that. Now, I do have insider information. So where is this, Moore's law for circuit size, this is really, from my perspective, the definition of SynBio. I have insider information, including projects in my lab, that we're at least going beyond this 6 regulatory unit limit plateau. And so I expect within the next year or so to have publications that have about 10 to 12 components in them. So I think there's at least a positive slope going on here. But that represents, an important thing to consider, as well as an important opportunity. So you have to identify what are the constraints, what's restricting our ability right now to proceed with synthetic biology, and then if you're interested, working on those things. Okay, and so I mentioned this notion of design. So I'll spend the rest of the lecture on that. So the idea is that we'd like to be able to, or at least those of us with a computer background, like to be able to think of some high-level behavior sitting in front of a computer, use computational resources to come up with a sequence, and then the DNA synthesis should be easy, right? So just press the print button, it goes to some DNA synthesis company, and it gives us the DNA in the mail. Right now it's about, you can get it down to about 25 cents a base. So it's not quite pocket change yet, but there's some indications that, again, as I mentioned before, it's going down and down and down. So you'll be able to really not do the DNA synthesis yourself. So hopefully, not for you guys, but maybe, in 5 years down the road, people will look back at this day and age where people doing molecular cloning by hand and go, oh my God, those people suffered so much. We have so much pity for them. And really, all of this would be, automated and cheap and so on. And then the idea is you get a piece of DNA, you put that into a cell, and then it does exactly what you want. Never happens, and so you really have to think about putting the DNA into the cells, finding out what's going on, and then iterate over this process. Do this debugging process that we mentioned. Okay, so in terms of the parts, so when we think about synthetic biology, we think about these parts. So I showed you how to create the DNA sequence for those. Okay, so in terms of the parts, what are those parts? So we want parts that regulate. So this could be a piece of DNA. So you regulate another piece of DNA. You can regulate messenger RNA. You can regulate protein-protein interactions. Cell-cell communication is another class of important parts. Debugging was also used. So these are bacterial cells that have been engineered by one of the iGEM teams in Cambridge to make dyes with different colors. So you can visualize what's going on in there. Okay? And then you want to interface with a cell. So you want to sense what's going on. You want to be able to affect what's going on in the cell. So you can think about this, this is the library of parts that we want to be able to use. Okay? So let me show you. So this notion of having this book of parts, TTL data book. Anybody heard with this TTL data book? Nobody here? Well, so this was really popular in the 1960s, where this notion that you have this book, this is pre-internet, that did exist at some point. So you have this book which you can buy and you can flip through. It has these parts that are described. And you can say, I want this part, this part, this part, this part. You put it together. Okay? And so now we have the registry. I mentioned the registry of standard biological parts. It's organized, it's accessible by parts. You have these parts, you have devices, systems, and so on. And you can get these, and put them together. Right? So it's all online. You can do your searches and so on. Okay, so what does a part look like? So here's probably one of the most basic parts. And so you can think about this part as doing a digital inversion. Okay? And so the idea is that we want to be able to regulate activity inside the cell. Okay, so this is a pretty simple part. So we're going to represent protein concentrations. They're going to represent signals. So you're going to have one input protein. And so if it's absent, that means it's a 0. And then we want the output protein to be at a high concentration. So these are going to be different proteins. And the idea is that if the input protein is not present, it allows a production of the output protein. So you have this molecule RNA polymerase, you have this process of transcription and translation, which results in the production of the output protein. Okay, so this is a very simple, basic process inside the cells. And then when the input protein is present, it represses the production of the output protein. Right? So in principle, the question is, can we take this simple process and have this 0 to 1, 1 to 0 mapping? Right? And the thing that's critical about something like this is that that's really the basis. So this digital inverter is really the basis of computing. So if you have something like this, and actually if you're able to take something like this, connect it, and build slightly more complex circuits, complex devices, and then build circuits out of them, devices like the NOT AND gate, the NAND gate, NOT OR gates, then you can build anything, basically any logic function that you want. Right? This is the basis of computing. And so the question is, if we can have something like this, can we actually build anything that we want inside the cell? So we want to have something that has basically this inversion, ideally in this kind of function. So this would be, so again, we're dealing with protein concentrations. That's something analog, right? There's some kind of analog concentration of that protein inside the cell, but we want to still have digital behavior. So how is that achieved? So actually, by the way, that's the same in electronics, right? So in electronics, you have these analog voltage values that are then somehow processed from input to output so that we always have a digital, basically meaning a digital operation. So it's always pretty much high and low, high or low. Okay? So the way you'd like for this to work is that we have this input to output relationship where if you have a 0, something that's analog in this range, then it gets mapped to a really nice looking 1 over here. And if you have a 1 that's a kind of noisy, then it gets mapped to, on the output, gets mapped to a really nice looking 0. This is called signal restoration. right? That's what we'd like to see out of the behavior of our system. So this is not possible in reality, okay? But in biology, so in electronics, this is not even possible, but we get something close to it. In biology, we also, the question is, can we get something close to this? And so now the key is, if you have devices that have these properties, then even if they're a little bit noisy, but they still have signal restoration, and you put them together in sequence, the key is that as the systems get longer and longer, they still maintain this digital operation because of the signal restoration. They still pretty much work, the outputs still pretty much work at zeros and ones. Right? And that's again the key for electronic computing. Is the fact that even if you have these basic electronic units and there's a little bit of noise in the system, each element cleans up the noise and allows the digital operation to continue. Okay? And so let me skip this one. And so in my thesis, I was first characterizing this, what's called the transfer function, this input to output relationship. And then later on, when I went to Princeton, I asked the question, can we then take these noisy biological components, put them together in series, and would they give us digital behavior? Right? So this is a really important question. So if the answer is no, you put a lot of these pieces together and they just gets worse and it gets noisier and noisier, then we're kind of in trouble. Right? It's going to be very difficult to expand to get to larger systems. And so here we have a sequence of basically inverters like the ones that I showed you. And the question is, what happens to the output as the system gets longer and longer? And so let's just focus on the steady state behavior. This is input versus output. And what I'm showing you here is the blue is the first stage, the black is the second stage, and this orange-yellow is the third stage. And what I'm showing you is this is input to output mapping, that as the system gets longer, it actually becomes more and more digital. Okay, so we're really excited about this because that means that these noisy biological components actually have the right properties if we engineer them properly so that we can build large-scale systems out of them. Okay, so it makes sense? Yes?
I agree with the fact that, in your analogy, that's sort of one stream at a time, right? Like single calculation or something like serial processing. Whereas in the cell, we probably have like a lot of different factors contributing to the same thing. So how do you deal with that?
So even in a computer, there's a lot of parallel processing that's taking place. So it's engineered parallel processing. It's kind of limited by our ability to design parallel processes. But if you think about an Intel chip, a quad processor, so it already is doing 4 things at a time, the quad processor. But even within the processor, there are many simultaneous activities that are taking place. There's things like pipelining and so on. So there's a lot of parallelism that's going on there. One thing that's quite different though, so parallelism is true for computer electronics. There normally is a clock. So normally things are synchronized. So even though you have parallel activities, they normally happen, every clock step something happens in synchrony, which simplifies our ability to design complex systems. In cells, there are actually clocks, right? So we work by clock. By our clock time is slightly different. So we have our circadian rhythms, for example, right? So it's a good thing we do more than one operation per circadian clock cycle. But so we have to think about other ways of designing biological circuits that are not synchronous at that level. Let me also point out that there's actually a field of computer electronics that deals with asynchronous design. So this is not common, but there's a subset of people that really specialize in asynchronous design where you don't have to have a clock, right? So you can build a whole bunch of logic gates electronically and then just get them to work together asynchronously. And so actually a lot of those people that are experts in asynchronous electronic design are now really interested in the biological substrate. Yep. So let me point this out. So what I want to do, I think for the second time, let me very quickly go through a few of these examples, and I'm going to skip over to a couple of these other examples that I mentioned. Okay, so I want to show you very quickly a few simple modules. So here's another one. This is a 1-bit memory, basically. Okay, so the idea about this 1-bit memory is that you have these two repressors, two proteins that repress each other. So it's basically like that inverter that I showed you. This is essentially an inverter with the ability to modulate it from the outside. So you have two repressors, they co-repress each other. And that means that basically only one of them should be dominant at one time. Okay? And you can then flip the switch externally. So the idea is that if you add this molecule called IPTG, then you're able to basically turn it on. Okay, so turn it on means that this C1, the lambda repressor, also has a green fluorescent protein associated with it. So you add IPTG to the cells, it gets turned on, GFP gets turned on, you remove the IPTG, okay, it's no longer there, the cell remembers. So it actually has memory. And how does it remember? Well, even though there's no IPTG there, the GFP remains high. Okay, these are on the order of hours. Okay, then you can incubate it at 42 degrees. That's a way to flip it back, reset it, and it gets reset. So it goes to 0, and then you leave it there, and it remembers the fact that it was reset. Right, so this is a 1-bit memory, and the critical thing here is it has hysteresis, so it remembers. So here, in this region right here, and this region right here, it's exposed to no inputs at all. Right, so it has the same environmental conditions, but its own internal state is different, based on whether you told it right before to set it or reset it. Okay, so this is the basis of some additional digital logic that you can put inside the cells. Let me skip this. You can do, just a point I want to make, it's not about just digital. There's actually a lot of emphasis about analog devices. So how do you program a cell? Is it like the way you program a computer? Not exactly. Is it always digital? No. So it's going to have some digital control. It's going to have some analog control to it too. So this is just showing that we can do analog control here as well, quite precisely. This is an example of a clock. Okay, so taking these analog mechanisms, I'm going through this very, very quickly. So I'm not explaining to you the details because I want to get to those two other examples. But if you do have a question, if you want to talk about this in more detail, I'm glad to stop and give you more detail. This is now a clock that was made in bacteria using cell-cell communication. So cells send a signal to their neighbors, and when they receive that signal, they make more of it, so it's a positive feedback loop. And at the same time, they make an enzyme that degrades it. So there's a fast positive feedback loop and a slower negative feedback loop. And when you combine those together, that's actually, by the way, this fast positive feedback coupled with a slower negative feedback is the basis for most clocks in cells. Circadian rhythms, for example, work based on this operation. Many clocks in biological cells have this kind of motif. So this actually, the design of the system is inspired by biological analysis. Okay, so it's looking at what works in biology and then trying to make it actually work in a synthetic biology context, actually, but modifying it slightly to also include the cell-cell communication. And so what happens is you have a community of cells. Let me show you what it looks like. So this is in a microfluidic. These are all individual bacterial cells here. And then you see initially not so synchronized, and they communicate with each other, and they're all really, doing this clock. They're fluorescing in synchrony, which is actually pretty cool. Here's another movie. So you see the bacterial cells, and then you have all these kind of shooting rays, which are kind of fun. Okay, so you can really, have fun with all these. This is actually part of the reason people get into synthetic biology. They want to, make cool things under the microscope. Here's something else. There's something that we did in my lab to get cell-cell communication. So this had more transient properties. And this what I showed you was done in 2010. This is work that we did in 2005. So the idea is to get cells to talk to one another. So again, because of time, I won't get into the details, but cells send messages back and forth, and they interpret, they process these messages, they look at the analog values of those, and they decide to make fluorescent proteins based on those analog concentrations. Okay? And so we have sender cells that send out messages, we have receiver cells that respond to those and make different colors. And without going to all the details on how it works, this is the experimental setup. We have initially undifferentiated cells. We put sender cells in the middle, and we have these cells basically respond to them. And so we can make patterns that look like this, basically programming the bacterial cells to communicate and then make patterns. And this is a variety of different patterns that we're able to make in bacteria. And so now, so we have this initial ability to create patterns by design. And now we're doing this also in mammalian stem cells. We don't have, quite a working heart in a cell culture, but we're taking the basic steps towards programming mammalian stem cells to talk to one another, create three-dimensional patterns by design, and then based on these patterns, make decisions to differentiate. So, for example, the green ones could become bone, the red ones could become neurons, the yellow ones could become, other cartilage or things like that. All right, so the question is, can we program these stem cells to differentiate to any kind of tissue that we want? And this is, by the way, related also to the work that's going on in EBICS, which I'm sure you've heard about, in terms of how do you create biological machines. So my take on it, and now going on and interacting with all the folks in EBICS, which has been tremendous, is I was mostly initially focused on cell-cell communication and information processing and so on, but now also interaction with the physical aspects, the cell motility, cell matrix interactions are really important. So taking all of these factors into consideration when creating these tissues by design. And really, it involves getting expertise from many different folks and bringing them all together.
And remember that for when I speak next, because that's exactly where I'm going to pick up.
Okay, great. Okay, so let me. So the idea would be to have a high-level specification and figure out how to engineer individual cells so that they interact with one another locally. So eventually, they create this high-level behavior. Okay, so what I'm going to do now, we have 10 minutes left, so I'll skip to two applications, cancer, and also, I'll see if I get to this diabetes thing. So we're working on automation. I mentioned this notion of automation. We'd like to be able to write high-level programs and then have compilers that just generate these instructions so that a robot like this, so this is a robot in our lab, so this, hopefully in the future this will replace, I don't want to say replace graduate students, So it's not like you're going to be able to leave earlier, right? Your PhD is not going to take you 2 weeks. It's just that we're going to expect a lot more out of you. So this is when my graduates say, "Oh yeah, I can graduate much faster." No, that time is fixed. We just expect you to do that. So yeah, but the cool thing is that the graduate students and the postdocs will be able to focus kind of on the high level stuff, on the design, which I think is really where the cool parts happen. And so this is, by the way, so this, we've gotten the robot to do most of the assembly for us already. It's much easier to program a real robot than a biological robot. So I'm certain that we can get this fully automated, this within two months. There's nothing really fundamentally that difficult about it. Once you build the DNA, who knows what's going to happen inside the cell? That's much more difficult. So this is, so using compilers, what's the computational design tool of your future going to be? Are you going to write just computer programs? Well, sometimes yes, but sometimes you might, what would the computer design, the visual design be for tissues, for EBICS, right? So rather than something like this, we might use tools that look like this, but then allow us to also encode function in it. right? So you can visualize, I want to put, the pumps over here, that somehow the muscle is connected to, the vascular networks over here with neurons are connecting them everywhere, and you can kind of, visual basic functions that you can put in there and think about the overall structure. So this would be kind of a cool world to be in, where you can just think about these, biological machines or tissues and build them like this, and then get the liquid handling robot to build the DNA for you. So with the remaining time, I just want to talk about, I guess I'm mostly just focused about cancer. So tissues by design is one application that I'm really excited about. Another application is, if we can get, and this tissues by design is, I would say, a long-term goal. The notion of using synthetic biology for cancer therapeutics, interestingly, I think is actually a shorter-term goal. Okay, and I'll try to explain why. And so here we're trying to address this notion of one of the important challenges in cancer treatment, which is specificity. So you can have a therapeutic agent, which, the idea is to have a therapeutic agent which will go and kill the cancer cells. So you can have a therapeutic agent like that kills all the cancer cells. Problem is, at the same time, it's probably going to kill the patient as well, right? So we don't have. So for example, if you kill all the cells that are dividing, that will kill all the cancer cells, right? But that's a big problem. That's oftentimes going to result in the patient actually dying. That your body just can't. That's not how you can function. And so if we can get something with synthetic biology that can give us that specificity, I think that would be a huge advance. So basically, almost completely, it doesn't have to be completely, but almost completely eliminate false positives and false negatives. Okay, so what's the idea? We'd have a therapeutic agent that, so for example, right now there are these agents that will look on the cell surface and try to make a decision about whether the cell is cancerous or not. But even if you look at a single marker on the surface of a cancer cell, that's typically not enough to tell you whether that particular cell is cancerous or not. Okay? So the idea is that this agent goes inside the cell, and then all the information that you need is inside the cell. Right? So this therapeutic agent basically integrates multiple pieces of information inside the cell, and then makes a decision, which should be highly informed about whether to kill the cell or not. And by the way, even if this is not the cure for cancer, obviously I'm not going to stand here and guarantee that. There are many non-clinical applications for this. So even just this notion of being able to have an agent that goes into the cell and tells you what's going on in the cell, that integrates multiple pieces of information and gets a cell to fluoresce in different colors based on that, that's going to have huge number of applications in biology in general. Okay, so the idea is that we looked at cancer cells from HeLa cells. Anybody read this book about Henrietta Lacks? Okay, so this was in the New York Times bestseller. I haven't actually read it. Is it good?
Not really.
Yeah, that's what I heard.
Too much story.
Yeah, so I got my father-in-law to read it, and he said, So, anyways, but I still put it up there. So this is actually this cervical cancer. So this is actually this cell now propagated, this cell type propagated. There's probably more of this HeLa cell than any other cell in the world. But the idea is that, again, we'll have this therapeutic agent which we can uniquely identify HeLa cells versus all other cells. So what's the idea? So the high-level behavior is detect and kill HeLa cells. Here's a program. Okay, so this is the program that can distinguish that. This is a little bit ugly here, uses this notation, but the program is actually rather simple. Okay, so the idea is that these biomarkers have to be high, and these biomarkers have to be low, and that's a HeLa cell. Okay, so that's the hypothesis. And we base this hypothesis on bioinformatics data looking at our markers, which are microRNA. So we looked at what's known about microRNA levels, and we saw that if a particular cell has these microRNAs high and these low, that's pretty much a HeLa cell. Okay, so the nice thing about this is this is also a fairly compact profile. Right? This is hopefully something that we can build and put in maybe an engineered virus. So it goes into the cell and does its computation and then kills or not kills the cell. And the hope is that most cancer cells, and maybe even most cell types, would have just a sufficiently small profile, sufficiently small number of biomarkers, that if we can tell their levels, that's enough to distinguish that cell from all of the cell types. So we argue that this is a very generic way and general way to be able to target any cell. Okay, so the idea is that it would go, the circuit basically goes into the HeLa cells in green and then does the computation, kills them, or other cells, and when it goes to these other cells, basic computation says no, and it does nothing. Okay, so let me, in the remaining 2 minutes, I guess, show you how this works. Okay, how does this actually, what's the kind of quote-unquote magic behind this? So we need sensors, and we need a way to integrate sensory information. Okay, so we need sensors that respond. The simple one is a response when the microRNA, our biomarker, is low. Okay, this is actually a pretty simple sensor. Okay, so we have a piece of DNA, we have messenger RNA that gets made, and then microRNA is this small molecule inside the cell that degrades messenger RNA. Okay, and so what we did is, this is endogenous. So this is microRNA. These 6 biomarkers were endogenous, right? And so if the microRNA, so what we did is we created this output messenger that we synthetically put these microRNA target sites on, such that if this microRNA endogenously is high, then it degrades this and the output is low. And if the level of this microRNA is low, then the output is high. So that's how you build what we call a HeLa low sensor. It's like the inverter that I showed you, in a sense. But it's sensing the microRNA that's endogenous to the cell. Okay, and then you can, the nice thing about this is, in a fairly straightforward fashion, you can create a multi-input logic gate. Okay, and so here the idea is this is basically an AND gate of inverters. Okay, meaning that the output is high only when all the inputs are low. Okay, so it has to be the case. If any one of these inputs is high, that's enough to degrade the output. So this is basically like the AND gate of NOT. Okay, so that's how you can build a multi-input logic circuit. So how do you build? So that's how we get the AND of the low sensors. How do you build a high sensor? A high sensor meaning that when the input is high, you want the output to be high. So what you do is you connect this low sensor essentially to the inverter that I showed you in the beginning. So basically, to get a high sensor, you do not. Okay, and so that's the way it works. So that you could do the not operation. So it's a needs repress this repressor. This repressor is actually the same repressor that represses the final output over here. So, but it has to get repressed both from here and from here in order for this output to actually be high. So that's how you create a multi-input AND gate that responds to basically high sensors. Okay? And so you take this, so that's the high sensors, you add this, so you can fuse them together, and then you get this entire logic function. Okay? So I went over this a bit quickly because of time. This is, now you see this is pretty complex. Right? This is something that you think about, synthetically, we have to create all these interactions, we have to get everything lined up, but it does work. And so, without going into all the details, we get this to work. It does recognize HeLa cells actually versus other cancer cells. So it's even cancer type specific. It'd be okay if it killed other cancer cells. So we didn't really design it. We designed it to be to have it distinguish between HeLa cells and healthy cells. If a byproduct that kills healthy other cancer cells, no problem. We won't get sued over that. But we wanted here to demonstrate that it's the specificity, as opposed to just the general cancer killing. And it does work, and it does not just fluoresces in the right color, but it also kills. So when we put a killer protein, it will kill this is all in cell culture, it will kill the HeLa cells and basically not really harm the non-HeLa cells. Okay, so now we're working on taking this from, a proof of principle concept in cell culture to trying to understand, what is it going to take to actually make a therapeutic out of this. And as I mentioned, and this is, I guess, where I'll stop, that this, general ability to build logic circuits that can sense information and then can actuate, killing the cell is one actuation that typically, people like to do. But there are other kinds of actuations. So, for example, differentiation, right? So sense what's going on in the cell and then carry out a differentiation process, so on. So as I mentioned, this could help in many other, so this notion of tissues by design is one example where it can help. So with that, I will stop. I don't know if I get booted out of here. Whether
Will you be around to answer questions during the break?
Yeah, so I can hang out during the
You're
break.
welcome to come back for lunch if you'd like.
Okay.
It's in
Yeah.
314.
Okay. Yeah. This is kind of my prediction, so I'll leave it out there. But if people want to look at that.
Leave it up during break.
During break. So if people are interested in the future, not guaranteed.
Thank you.
Yeah.
So we'll take a 15-minute break.